this video, I'm going to be talking about NK cells, or natural killer cells, as well as the NKG2D receptor found on the natural killer cells and what exactly that receptor does. Also, there is a compound or compounds that influence related pathways like PGC1A or immune activity, and I'll be talking about PGC1A as well. So the main role of the NKG2D receptor on NK cells is to detect stressed, infected, or cancerous cells. And usually there's a recognition of the target. NKG2D recognizes stress-induced ligands such as MICA and MICB or MICA or MICB, and then ULBP family proteins. These are upregulated on tumor cells, virus-infected cells, and cells under oxidative or DNA stress. So those ligands on the tumors and virus or infected viruses and cells under oxidative or DNA stress, those ligands act like danger signals. Okay, but the NKGD2 cannot signal on its own. It must bind to the DAP10 or DNAX activating protein of 10-KDA. That's a really long, fancy title, so DAP10 is a lot easier to say. So the key roles of DAP10 is that it acts as a bridge between receptor and signaling pathways, and the pathway activated is the P13K pathway. So DAP10 recruits P13K, which leads to PGC1A activation, or PGC1A is related to this DAP10 and NKG2D. So the things that can make the tumors express the ligands that the NK cells see as a danger signal is triggered by radiation, chemotherapy, which has negative effects like weakening the heart and muscle. So chemotherapy might not be a good way to make tumors more visible to NK cells. Also, replication stress. And then the key pathway is ATM, ATR, and P53 partially. So this activates transcription of MICA and MICB and ULBPs, which are the ligands on the tumors that NK cells have to detect so that the NK cell can attack and induce programmed cell death or apoptosis on the tumor. So there are tumors, they try to hide these ligands, MICA, MICB, etc. And they do this very actively as a tumor evasion strategy. So it can even shed the ligands with metalloproteases such as ADAM10 and 17, even released as soluble ligands, and can even downregulate NKG2D on NK cells. A second way is exosome release. Tumors export ligands and vesicles. So without the exosomes, then the tumors can't really do all of these things. So if you've already seen my other videos on exosome formation, MVB sac formation, lysosomal membrane peroxidation, etc., then you already know what I'm talking about. If you don't, then you should definitely check out those videos, because then this video will make a lot more sense. So another way besides exosomes, the exosome is probably the worst way that tumors evade NK cells, but they also hide those ligands, MICA and MICB, by internalizing, because this keeps those ligands inside instead of on the surface, because if they're on the surface, then the NK cells can see the tumor. They will see the ligand as a danger signal, or more like an eat me signal, and then attack the tumor. Okay, so another way that NK cells and also T cells, so they attach on the target cell. So the target cell, probably a cancer cell, has ICAM1, ICAM2, and then the NK cell with the integrin, or the LFA1, binds onto the ICAM1, ICAM2 on target cells, stabilizing contact, allowing immune synapse formation, and then apoptosis with the granzyme B perforin. So IFN-γ can boost VCAM, ICAM1. It's a key immune molecule that helps activate macrophages and strengthen antiviral tumor defenses. So foods that can improve IFN-γ are protein-rich foods with amino acids like chicken, turkey, fish, salmon, tuna, eggs, legumes such as lentils and chickpeas, and Greek yogurt. I tried finding something to boost LFA1, which is the integrin that binds onto the VCAM ICAM1, but as of up to date, there is no evidence that shows or proves that foods can upregulate integrins or LFA1 on NK cells or T cells. Okay, now on to PGC1A activation. Chocolate could be probably the best one for PGC1A because what chocolate does is that it boosts PGC1A mildly so that there is still mitochondria, healthy mitochondria, and then balanced ROS, and then regulated NKG2D ligand expression on T cells, so control the NK activation. But the problem is that if PGC1A is too high, then that could possibly reduce NK cell targeting, because PGC1A deficiency or being lower induces mitochondrial stress, increased reactive oxygen species, and then upregulating NKG2D ligands, and then enhanced NK cell targeting. So lower PGC1A seems to be better for NK cells. So chocolate boosts PGC1A mildly. Now, there are other foods that can boost PGC1A even more, like, for example, salmon combined with olive oil and exercise. All those three can really boost PGC1A, but that could go too high to the point where there's too much PGC1A, and then the NK cells lose their enhanced NK cell targeting capability. Okay, so it has been shown that if PGC1A like skyrockets too high, the NKGD2 ligand expression on the NK cell, as I talked about earlier, can actually be downregulated, or there is less expression. So that means that the NK cell cannot bind onto or recognize the tumor. Also, tumors, they try to stop mitochondrial rupture in order to stop NK cells. Because what happens when the PGC1A goes too low, or PGC1A deficiency, there is a mitochondrial rupture, and then that actually boosts or enhances NK cell killing activity. So the tumor tries to escape immune attack by stopping mitochondrial rupture, which is actually beyond the scope of this video. So other foods that are good for NK cells are astaxanthin in salmon, supports mitochondrial health, PGC1A activation, NK cell activity. Also, fermented foods like kimchi and sauerkraut can indirectly enhance NKGD2 responses on the NK cell. Also, NK cell activation. So that's definitely great against cancer. Okay, this is where you have to be careful to not boost PGC1A too much, which is because of exercise, especially endurance and HIIT, also HIIT, also fasting or calorie restriction and cold exposure. These are probably the strongest activators of PGC1A. Now, be careful not to do that too much, because then excessive PGC1A will end up leading to the NKGD2 receptor on the NK cell being downregulated, and then it cannot attack tumors. So that's it for the video. If you're interested in more future videos, then make sure to like, comment, share, subscribe. Keep it locked in here on this channel, and I'll see you in the next video.